Dear students, today we will discuss Unit 5 Basics of Phytochemistry, Applications of Electrophoresis in Isolation, Purification, and Identification of Phytoconstituents. Myself, Dr. Raju Wadekar, Assistant Professor, Shri Vile Pare Kelwani Mandal, Institute of Pharmacy. Disclaimer: this presentation is meant for educational purpose only. The content of the presentation is taken from e-sources as well as from author's learning experience and subject knowledge. The contents for this today's topic, applications of electrophoresis, is as follows: introduction, types of electrophoresis, prerequisites for electrophoresis, techniques, instrumentation, applications, and references. Now we need to understand what is electrophoresis. Electrophoresis is a method wherein charged molecules in solution, chiefly proteins and nucleic acids or amino acids, migrates in response to an electric current. So with this basic principle that all these proteins and amino acids, nucleic acids which carry anions and cations in their chemical structure, they give response to the electric field which is provided externally, and thereby they induce separation of these different chemical entities, proteins, amino acids, and nucleic acid. Electrophoresis is also known as cataphoresis. This electrokinetic phenomenon was observed for the first time in 1807 by Rus from Moscow State University, who noticed that application of a constant electric field caused clay particles dispersed in water to migrate to the anion and respectively cations. Thereby, it is ultimately caused by the presence of charge interference between the particle surface and the surrounding fluid. So, it is a very simple concept that after applying a proper electric field, a measurable electric field to a liquid containing a anionic and cationic mixtures, after applying that electric field, these electric fields induces separation of the respective anionic cationic containing proteins and nucleic acid to their respective interference. Okay, so anionic will go to the cationic molecules will go to the anionic electric charge current. Factors governing the migration of ions. Charge of ions is very important. One, if the charges of ions they are more or less, accordingly the separation or migration of ions will take place in the solution. Size of the ions, viscosity of the solution containing ions like cations and anions, voltage applied, the frequency of voltage depend on, and that the current, what voltage means here, the current, electric current which we are providing. So based upon the amount of voltage of current or the we are applying, based on that the separation of the compounds will take place. Similarly, pH of the buffer and ionic strength of the solution will also have direct impact on the migration of ions in the solution. Factor affecting electrophoresis are mainly molecular characteristics like size and shape of the compounds, buffer properties, electric field, and temperature. As an analytical tool, electrophoresis is a very simple, rapid, and highly sensitive technique which induces or which facilitates a proper separation of anionic and cationic compounds. And as you can see here, the electric double layer is there, okay, and now the power is on. So whenever the compounds are a mixture of compounds contain anionic and cationic compounds, they are separated and then through the electric current, okay, and then they are detected. Each of these anionic and cationic separated compounds are detected and then they are easily separated with the help of electric current. It can also be used preparatively as a separating technique. It can be used analytically to study the properties of single charge species or a mixtures of the molecules. Now we'll study the types of electrophoresis. There are total 12 different types of electrophoresis. These electrophoresis are classified, or the types are based on the functionality of each one of these electrophoresis technique. It means if at all the electrophoresis, if at all we are using a paper as a compound or as a stationary phase, then in that it is also called as paper electrophoresis. It is also the second type is affinity electrophoresis, capillary electrophoresis, third one, fourth is dielectrophoresis, fifth DNA electrophoresis wherein the DNAs are separated, gel electrophoresis means where the gel is used as a stationary phase, electroblotting technique, electrofocusing technique, immunoelectrophoresis, isotachophoresis, protein electrophoresis, last but not the least, pulse field gel electrophoresis. So based upon the base which we are using, based upon which technique we are using for separation, or what type of current we are going to use, which type of detectors we are going to use, which type of sample we need to separate or isolate and identify the phytoconstituent, the nature of the phytoconstituent. So based on that, the types of electrophoresis are available and you can opt any one of this method for separation. Now we will study in detail about each of these methods and its specific characteristics and application. Number one, paper electrophoresis. This technique is useful for the separation of small charged molecules such as amino acids and small proteins present in the plant material. Strip of filter papers is moistened with buffer and the ends of the strip are immersed into buffer reservoirs containing the electrode. So here a simple strip of filter paper is moistened with a buffer solution and the end of the strips are immersed into buffer reservoir containing electrodes, both anionic and cationic electrode, positive and negative charge. The samples are spotted in the center of paper. High voltage is applied and the spots migrate according to their charges. After electrophoresis, the separated components can be detected by a variety of staining techniques depending upon their chemical entity. It is same as that of thin layer chromatography. The only thing is the separation is take place based on the electromagnetic field or say electric field which we are applying to the mixtures, and based upon the anionic and cationic components affinity towards the anionic and cationic spots, they are separated and then they are further detected by staining procedure. Affinity electrophoresis. The methods are based on change in the electrophoretic pattern of molecules through biospecific interactions or complex formation. So, this method has been used for determination of constants. Some compounds which are having very constants or very close affinity towards that particular anionic and cationic electrodes. So, based upon their quantification, the alternative quantification of the proteins or amino acids can be done. Especially plant proteins and plant amino acids can be done. Separation of this compound can be done. Facilitates by affinity electrophoresis. Third one is capillary electrophoresis. As the name indicates that it is known as capillary zone electrophoresis. These can be used to separate ionic species by the charge and frictional force and hydrodynamic radius. It can be performed in capillary formats. So capillaries are mainly utilized as a stationary phase. The typical system consists of two reservoirs and a capillary filled with buffer solution. A high voltage is applied across the capillary by using a high voltage power supply. The very small diameter capillaries, like 5 to 100 micrometer, employed in this technique allow for sufficient heat dissipation or separation of compounds. Further, the fourth method is called as dielectrophoresis. It is a phenomenon in which a force is exerted on a dielectric particles when it is subjected to non-uniform electric field. Means, say for example, if a mixture in which the force is further exerted where the electric field is non-uniform, if the electric field is not properly going not it is going through the uniform pattern, then one has to provide external force on a dielectric particles, thereby it facilitates the proper separation. All particles exhibit dielectrophoretic activity in the presence of electric field. However, the strength of field depends on the medium and particle and on the particle shape, size as well as the frequency. See, every time whenever applying any one of these method out of these 12 methods of electrophoresis, one need to very much sure about the size, shape, electric field which you are providing, the frequency of electric field. So if they are all in a proper proportional, in proper ratio, then only it will facilitate proper separation of various ionic and cationic compounds. Fifth one is delay electrophoresis. As the name only indicates, it is mainly used for separation of DNA fragments, again based on their size. The DNA molecules which are to be analyzed are set upon a viscous medium. So viscous medium on which the DNA medium or the DNA compounds or molecules are placed, and the gel where the gel where electric field induces the DNA to migrate towards anode due to the net negative charge of the sugar phosphate black backbone of the DNA chain. Definitely yes. So the separation of these fragments is accomplished by exploiting the mobilities with which different size molecules are able to pass through the gel. So the gel, the viscous medium on which when we are applied a mixture of plant protein or plant DNA, and if I want to see different DNA molecules present in that plant mixture, then I just have to provide a small electric current at the specific frequency. Then I can see that towards the anode only all that DNA molecules are get migrated because the negative charge of sugar phosphate backbone, and thereby it induces, it elicits a proper separation of these compounds. Whereas gel electrophoresis, the number 6 one, it refers to using a gel as an anti-convective medium. In very first case, we have seen paper as a medium for separation. Here, the gel is used as an anti-convective medium or sieving medium during electrophoresis. Means what exactly is happening here? If you can see here, I have just drawn here. So this is a plate on which the medium is poured. Maybe any gel, it is a agar, okay, so agar gel is being used. Now if I prepare a proper sieve here with the help of a particular blade or say knife, then I can get the hooves over here, and these hooves help me the proper separation of the compound. So this is a whole gel mixture on which I have to apply the sample, and then the electric current is passed, and therefore it further facilitates the separation of major biological macromolecules of plant DNA, RNA, and proteins. It refers to the movement of charged particles in an electric field. Electroblotting, which is a seventh type of electrophoresis technique. It is a method in molecular biology. Biochemistry is mainly employed in immunogenetic to transfer proteins or nucleic acids onto a membrane by using nitrocellulose. Again here, instead of using gel or say paper or say any viscous medium, here we can use a nitrocellulose membrane. So after gel electrophoresis, we can simply use a nitrogen membrane. These are the inert compounds, and they facilitate very nice separation of such type of biological molecules or biomarkers. The proteins or mononucleic acid can then be further analyzed using probes such as specific antibiotics, ligands like lectins or statins. Alright. The eighth type of electrophoresis is called as electrofocus. Electrofocusing is all about isoelectrofocusing, also known as electrofocusing, is a technique for separating different molecules by the electric charge differences. It is a type of zone electrophoresis usually performed on protein in a gel that takes advantage of the fact that overall charges on the molecule of interest is a function of the pH of its surrounding. So it is mainly based upon the change in the pH of surrounding will change the separation of the molecules, the charged molecules from the mixture of compounds, and thereby you can easily separate such mixtures of compounds which are more pH dependent compounds. So though we are providing the sufficient and effective electromagnetic fields or electric field, but still it is not, you are not being getting the proper results. So it is due to the pH dependent factors. Therefore, electrophoresis is one of the technique where we can separate the proteins on a gel in a gel, and that takes the proper advantage to the fact that overall charge on the molecules of interest is a function of the pH of its surrounding. Ninth method is called as immunoelectrophoresis, which is also called as gamma globulin electrophoresis or immunoglobulin electrophoresis. It's the method of determining blood vessels of three major immunoglobulins, blood levels of three major immunoglobulins like IgM, IgG, and IgE. It is mainly employed in biological fluids analysis, whereas isotachophoresis, which is a 10th type of electrophoresis technique. This technique employs the analytical chemistry used to separate charged particles. It is further development of electrophoresis. It is a powerful separation technique using discontinuous electric field to create sharp boundaries between the sample constituents. So means after this continuation of this electric field, still you will get the proper separated compound separated between the sample constituents. So they will not go and mix with other sample constituent, but instead they will have a proper, proper separated boundaries in the given field. 11th method called as serum protein electrophoresis. Because of the protein in monoclonal, each molecule has identical electrophoretic qualities that moves it to the same place in the electric field, creating narrow, intense band or spike. So we need to understand this. Say, if the proteins are of same structural significance, then you may get lot of the accumulation of the same type of protein structures at one site. So that should not happen, and therefore the serum protein electrophoresis is mainly employed. So the discovery of M component suggests the presence of multiple myeloma monoclonal gammopathy of undetermined significance, and Waldenstrom's macroglobulinemia, other lymph proliferative disease, or primary systemic amyloidosis. So these components, especially the protein spikes or the narrowing and intense bands, may happen due to the proteins which are being detected are of having same intensity, same electric field, same separation pattern. So that should not be there, and therefore the M component suggests the discovery of M component type of serum protein electrophoresis helps in the proper identification of various myeloma monoclonal gammopathy of undetermined significance, where it is used to treat to diagnose a disease. Last, which the electrophoresis technique is called a pulse field electrophoresis. It is a technique used for the separation of large DNA molecules by applying an electric field that periodically changes direction to a gel matrix. And the theory behind why PGE works pertain to the mobility of large fragments. So, because this type of pulse field, so this pulse field electric field will give the proper separation of major large molecules of DNA molecules of plants as well as of human genome, and thereby it will facilitate proper separation of these large molecules of DNA, and thereby gives a proper separation of proteins, nucleic acids, and DNA. Now, dear students, I just keep on telling you through this presentation that there is separation of proteins, nucleic acids, and DNA take place for the plant. So, what is the main application of this electrophoresis? Is a complete and not only isolation of the DNA and the protein and the nucleic acid or amino acid, but also its purification, as well as a complete identification of the specific type of protein present in the plant material or provide an isolated compound. Say, for example, if an alkaloid is there, so if there is a mixture of alkaloids, then from that alkaloids I can see the amino group is N plus. So it can easily get separated or they identified or purified from the mixture of compounds through the electrophoresis. And not only I'm separating, isolating or purifying this alkaloidal compound, specific class of alkaloid, at the same time I'm also doing its authenticity. Okay? So this electrophoresis in a very small amount of sample without degrading the sample, I'm getting the complete results for that phytoconstituent or the phytoconstituents or the mixture of phytoconstituents present in the sample. Now we'll need to understand the third point, that is prerequisites of the electrophoresis. The prerequisites means what are the requirements for these techniques. Mainly that you can see here now gel electrophoresis how exactly it works. The GIF is given here. So if the mixtures are there, so when it is applied on the gel, okay, this is a gel on which the mixture is added by applying electric field, okay, of a specific frequency. The different molecules mixtures present into it based upon the molecular weight, they are separated one after another in form of fragmented pattern, and then all that separated compounds are then further detected by, and then the whole information, electric information is given to the computer, and where in computer you can see here they are showing very nice separation of each one of that spot. Okay, and these spots or bands can be further quantified, and then it helps in the proper separation, purification, and identification. So the very first and foremost requirement is it requires a supporting medium such as Watson filter paper. It is same as that of your thin layer chromatography, paper chromatography, but here we are mainly employing a Watson filter paper, agar gel or agarose or cellulose or polyacrylamide as a stationary phase. Buffers are required for two different reasons. One is to provide the pH, which imparts charge on the resulting net charge, determines the flow of migration of solute to any one of the electrodes. Buffers like barbitals or trace acid EDTA are the most commonly used, which fix the effective pH at 8.6, leading to the sharper bands and good separation. In addition to above, the main important point is supply, power supply, positive and negative electrodes, chamber, and identification detection methods are needed for electrophoresis. So what you will need? You will need a proper supporting medium made up of Watson filter paper or agar or agarose cellulose, buffer solution to maintain the pH at 8.6, and of course power supply, positive and negative electrodes at both the ends. See, as you can see here, there is a positive and negative electrodes are there, and of course you will need a chamber where this whole process is carried out. Nowadays, they are so advanced that the detectors are also placed, or say they are it is integrated with that of the instrument, and then the detectors give the whole information to the computer that uses further separation and identification. Prerequisites for the electrophoresis further, we can see here polyacrylamide gel is can be used like this stationary phase. We can use them or sodium dosyl sulfate as a dissociating agent, and the rules in electrophoresis like polyacrylamide gel, synthetic gel, versatile medium for separation, agarose medium for separation, same as that of in TLC what we use silica gel. Okay, here sodium dosyl sulfate dissociating agent, bromophenol visualizing agent, glycerol as a preservative, because if you are using agarose, therefore there is always chance of contamination. So care should be taken that the medium should be preserved by applying glycerol and trihydroxymethylamino methane for satisfactory buffer in the pH range from 7 to 8 for the complete separation compound. Again, one more GIF we will discuss here, friends. Like, see, you can see the power supply, so which will be the positive and negative current. This is the base, okay, stacking gel. and then the sample is applied here, and then after applying the specific frequency of electric current, the separation of compounds takes place based upon their frequency and affinity towards the specific electrode, either positive or negative, and thereby you can get the proper resolution of the separated compounds in form of bands, and that they are further detected and then converted into a computer language where you can get into peaks or say bands, and then that compounds can be easily standardized with that of standard sample compared with the standard sample, and then they are identified very well. Again, here there is a schematic presentation of electrophoresis. As we can see here, a trough is there where you can put the buffer solution, and there is a cathode and anode, electric supply, apply your sample, load your sample here. So, and buffer solution on both the sides, upside and downside, and this is a gel on which the separation will take place. So, this is figure number 1 and this is figure number 2. You can see that these are the wells where you have put the sample, and then after applying the proper sequence of electric field, a positive and negative charge ions, the ions are separated, okay, based upon their affinity towards their respective electrons. The smaller fragments, larger fragments, medium fragments, and then further. This is the only basic principles where you can get the proper separation of, especially for proteins, amino acids which are present in plants, or say in various secondary plant metabolites and primary plant metabolites. Technique, as we can see here, we will require a base. So the base is nothing but agar gel. So place the agar gel in microwave, then you can liquefy it and then put it into a trough to prepare a base here. Then cool the mixture at 65 degree and pour into mold. Combine inserted, comb inserted into mold. Comb means, so after combing that, so you can get the proper separation. Or it's like a cassette or say well it's also we call it as. So once you do that, apply it, apply the sample, and then you after applying or after putting up electric field, supplying electric field, you can get the proper separation of compounds in form of a bands, and that they can easily be detected. An overview of the electrophoresis. The only thing is, students, you need to understand that the compounds which has affinity or which do contain some ionic or cationic compounds like proteins, nucleic acids, amino acids from the plants that can be easily separated, isolated, identified by electrophoresis. And you can opt any one of the method out of that 12 based upon your mode of application as well as mode of sample. Application to determine the mode of reproduction of plant species. To detect means we need to understand the mode of reproduction of plant species can be easily detected by electrophoresis wherein you require very simple small amount of sample. Also to detect the genetic variation within and between the plant population and to identify the plant genotype. So this is the complete selection authentication of the plant materials. This is required, very important in basic of phytochemistry. And further, the researchers establish the genetic relatedness in plants, that is, establish the most probable paternal parent or colon donor within and outside the study that seared seeds collected from a known maternal plant. So the very proper genus seeds should be selected based on these electrophoresis. So after doing electrophoresis of the specific genus like Sympona succeruda, Sympona legeriana. So if I'm using that Sympona species, first I need to understand the DNA, the protein, the nucleic acid present into it. Where first I have to do the proper separation, I have to standardize it, and then after doing that, after authentication done with the electrophoresis, I can very pretty sure that yes, the genus which I am going to use is of the proper maternal plant. Further, to accomplish this task, researchers rely upon protein and DNA markers generated by the gene electrophoresis to authenticate and identify the chemical nature of the isolated phytoconstituent. Also, to understand, it's very important whether it is alkaloid, whether it is glycoside, whether it is tannins or flavonoids or polyphenolic compounds. We need to understand that its chemical nature just by doing chromatography or spectroscopy. No doubt there we can relieve the complete, we can illustrate the structure. But again, to get more authenticity on that chemical nature, one can opt for electrophoresis as one of the fantastic technique for the identification of that phytoconstituent. Last but not the least, to develop the library for endangered plant species. Very important. By electrophoresis, one can easily develop a very nice library from its genetic makeup, from phenotyping, phenotypic makeup, and thereby we can understand, we can check the quality, purity, and identity of its genetic material, the plant species. Say, for example, if any plant is in the uh, endangered zone, then we can simply identify genes and proteins and nucleic acid, and that we can preserve for coming future years. So, in short, electrophoresis is a technique which separates, purifies, and identifies phytoconstituents or the plant materials and the plant DNA, proteins, nucleic acids, amino acids by the applications of the electric field, or by the principle that is after application of electric field of the set frequency, these molecules like nucleic acid and protein they depart, they can separate, they can identify on the given base. This is how electrophoresis is very much helpful in proper identification, proper proper utilization of authentication, identification of various chemical nature of isolated phytoconstituents. References, I have used these two different references, no doubt some of the some internet references I have used for the preparation of this topic, electrophoresis.